Welcome to my entry for the 2023 UCLA Undergraduate Research Showcase. Today, I will be presenting a set of experiments that aim to clone the Strongylocenteris purpuratus ADP-Ribosylation factor gene originating from a previously provided unknown sequence. Diving right into the heart of my presentation, we will begin by discussing some background information on the identity of the unknown gene, followed by the experimental design, goals of the project, the data and results obtained, and finally a big picture conclusion and any important takeaways, along with potential future research directions. The overall project goal was to successfully clone and determine the identity of an unknown gene from a predetermined and provided nucleotide sequence. Now, let's first review a brief introduction into the function of ADP-ribosylation factor family enzymes and the crucial roles that they play in organisms as a whole. ADP-ribosylation factor genes are responsible for catalyzing various types of post-translational modifications that are involved in a wide variety of biological processes, such as the detection of DNA damage, initiating DNA damage repair, and even cell proliferation, along with lineage differentiation. Now, ADP-ribosylation factors are members of the RAS superfamily of proteins and are specifically located in the Golgi apparatus with functions that mediate actin-based skeletal remodeling activity through catalytic control of GTP-GDP hydrolytic activity. Additionally, they are known to play a significant role in metabolic regulation, stress, and immune response, especially in the presence of foreign pathogens, and are very important in maintaining functional immunity in various organisms. Now, initially, to gain further insight into the potential identity of this unknown gene, the nucleotide sequence was subject to a bioinformatics query against the BLAST-N database. This revealed a high level of sequence similarity to a gene sequence that is found in the purple sea urchin ADP-ribosylation factor enzymatic family. To further confirm this, a phylogenetic tree, along with a translated amino acid sequence analysis, was conducted. After constructing a neighbor-joining bootstrapped phylogeny tree, the trend reflected expected traditional animal phylogeny, which provided additional evidence in support of the suspected gene identity indeed being correct. Furthermore, two distinct regions revealed a high level of amino acid sequence conservation and correspond to a GTP binding domain along with a P-loop NTPase binding domain when compared against known orthologs with similar sequences. These two domains are critical to the functionality of the ADP-ribosylation enzymatic family and provide an additional evidence that supports the hypothesis gene identity as well. The GTP binding domain is crucial for enzymatic activation and acts as a regulatory switch. In a similar fashion, the NTPase binding domain is responsible for the generation of a concentration gradient that alters cell polarity and thus changes the activation state of various additional secondary messengers, which ultimately have the potential to change and/or alter gene expression temporarily. After sufficient evidence was gathered in support of the gene's identity, we began the molecular cloning process. Thus, as a natural first step, we isolated and amplified the target ADP-ribosylation gene using a sequence-specific primer and employed the use of a GATA-E control gene to aid potential troubleshooting efforts if needed throughout the project. As depicted by the gel, a distinct band in each lane was observed and determined to be consistent in size when compared to the expected molecular weight of both the ADP-ribosylation factor 1 gene along with the GATA-E control gene. Thus, the amplification of the target sequences was successful and sufficient to proceed to the next step. After performing the initial gene-specific PCR-based amplification, both samples were subject to purification to remove any residual reagents that may be lingering around from the initial PCR-based reaction. As shown in the gel, the observed band sizes roughly correspond to that of the expected product sizes and thus were retained and not accidentally removed as part of the purification process. While the band corresponding to the ADP-ribosylation factor gene is faint, this is due to overexposure of the original image and is indeed present. Before proceeding, to ensure sufficient DNA concentration levels were present, UV spectroscopy was performed and revealed that both samples had DNA concentration levels that ranged from 37.3 nanograms per microliter to 48.7 nanograms per microliter. Additionally, the A260 over A280 fractional coefficient which indicates DNA sample purity, ranged from 1.7 to 1.82, indicating a seemingly high level of purity with minimal contaminants present. Thus, it was determined that the concentration of DNA present after the removal of contaminants was sufficient to proceed with the subsequent cloning process. Next, both purified PCR genetic inserts were ligated and incorporated into a stock plasmid vector prior to undergoing transformation into previously prepared bacteria host cells to generate a transgenic plasmid containing the desired target gene of interest. The use of a PBSK control plasmid was used to determine the competency of the previously prepared bacteria cells, ensuring that they retain the ability to uptake and upscale the newly generated transgenic plasmid. A standard blue-white selection technique was used to determine the transformation efficiency along with the experimental ligation efficiency, respectively. While the ideal transformation efficiency range is around 10 to the 6, our cells were 28% less competent than expected and were observed to have a transformation efficiency of 10 to the 5th. However, since the ligation efficiency of both DNA inserts fell within the range of 10 to 100 times lower than the transformation efficiency, this was determined to be sufficient to proceed with subsequent plasmid extraction. Next, we then used a standard mini-prep alkaline lysis protocol to isolate and purify the transgenic plasmids from the bacterial genomic DNA, along with any residual cellular impurities that may have been present. Ethanol precipitation of genetic material and subsequent plasmid DNA extraction using a phenylchloroform solution was used to separate the plasmid DNA from the genomic DNA by exploiting their vastly different molecular sizes. The plasmid DNA was then allowed to reanneal before subjecting 3 replicate samples to UV spectroscopy for concentration and purity analysis. The samples were determined to have a DNA concentration ranging from 2,125.2 nanograms per microliter to 2,603.7 nanograms per microliter, along with fractional purity coefficients ranging from 2.86 to 2.10, indicating that all samples were relatively pure with minimal protein or RNA contamination left over. Next, as a preliminary conformational method to ensure that the correct and desired DNA insert was successfully incorporated into the stock plasmid vector, a restriction digestion analysis was conducted and the DNA fragment sizes were analyzed using standard gel electrophoresis. The restriction digest process cuts the ligated pieces of insert DNA out from the plasmid vector, which in theory should produce 2 distinct bands corresponding to both the empty plasmid vector and the DNA insert size, respectively. Lanes 3, 5, and 7 all depicted bands consistent with the DNA insert product size as well as the empty destination vector, providing the first supporting piece of evidence that the correct DNA insert was indeed incorporated into the plasmid correctly. While the observed band sizes may show minimal discrepancy compared to that of the expected sizes, this is likely due to errors in estimation and are considered acceptable and normally expected. Additionally, it must be noted that the bright smeared bands present towards the bottom of most lanes is characteristic of residual RNA or bacterial genomic DNA contamination that was not successfully removed during the plasmid purification process, but regardless does not pose a substantial issue when interpreting these results. Furthermore, additional bands present in some samples are likely due to incomplete enzymatic digestion or varying DNA conformers such as supercoiled, open circular, or linear fragments, resulting in changes in DNA migration rates. Next, to gain additional evidence supporting the correct incorporation of the desired DNA inserts into the empty plasmid vector, both transgenic plasmid samples were sent for DNA sequencing. Upon analysis, the sequencing data returned was of high quality and compared against the nucleotide BLAST database. Both samples revealed a 98% match rate corresponding to the ADP-Ribosylation factor 1 gene, indicating the success of the molecular cloning project. Unfortunately, the SP-GATA-E control gene sample was mistakenly identified and instead revealed to be the ADP-Ribosylation factor 1 gene replicate, but this does not impact the conclusion whatsoever, as it was mostly employed as a troubleshooting tool throughout the entire molecular cloning process. Overall, the molecular cloning of the ADP-Ribosylation factor 1 gene was considered a success and confirmed independently through a restriction-mediated digest along with a more informative DNA sequencing analysis. Some potential follow-up experiments that can build upon the foundational research conducted throughout this project can involve subjecting the gene to various perturbation experiments to observe any changes in the organism when the gene is either overexpressed or knocked down, respectively. Furthermore, in situ hybridization could allow the in visual investigation of the ADP-Ribosylation factor 1 gene and has the potential to provide insight into any additional roles that it may play throughout the organism as a whole. Finally, here are my references, and I wanted to additionally thank both Pei-Yun Lee and Nick O'Connell for all their help and guidance throughout this project. Thank you for listening to my presentation, and I hope you enjoyed my project.